So record, share PowerPoint. There we go. Back to it. All right. So antibiotic spectrum, we have broad spectrum, so drugs that are effective against a bunch of different kinds of bacteria, or narrow spectrum, where we have drugs that are effective against a few types of bacteria or one category. So take a look at polymyxin and gentamicin. These are only effective against gram-negatives, as opposed to vancomycin and bacitracin that are effective only against gram-positives. So that would be a narrow as opposed to a broad spectrum. And we're going to call these secondary metabolites. So that is just a big word, meaning that it is a metabolic, so it is a biomolecule that does not help it grow or reproduce. So that's how we're going to define fitness, right? Getting back to our earlier lectures. So you got to survive long enough to reproduce. That is the primary metabolic investment. Survive, reproduce. Past that, we're going to say secondary. You want to be able to deal with your competitors. So we can have an antibiotic as a secondary metabolite. So we can do this to outcompete other species so that we can survive long enough to reproduce. Okay, so since we have all of these drug names to talk about, I've put these title slides in here to help categorize and organize this information a little bit more straightforward for you. So until we get to the next title slide, we're always going to be talking about inhibition of DNA synthesis. So all these drug names that we're going to talk about have to do with preventing bacteria from replicating their chromosome. And I'm going to also mention that all of these that we talk about until we get to antivirals are going to be antibacterial. An antibacterial agent will do nothing to a virus. I say that, and now I'm hesitant because I don't know every antibiotic that exists. There are some antibiotics that might be effective against viruses. This is biology. But just know that they are not designed that way. Okay, so one group of drugs, the sulfonylamides. So these are going to prevent folic acid from being produced. Folic acid is necessary for the replication of a chromosome, so for DNA synthesis. So that's what I want you to know. You don't need to worry about this PABA turning into folic acid, because again, this isn't biochemistry. Just know that folic acid is used in the process of DNA replication. So if you prevent that precursor molecule, we'll call it, it's a growth factor, then you're going to inhibit replication. You don't replicate, your cell can't move on to cause downstream problems in a host. All right, we have quinolones that are also going to inhibit DNA replication. These are broad spectrum. And then we also have another class called the, bear with me here, isoniazid. Yeah, I think that's probably the hardest one to pronounce here. And so this is going to be effective against mycobacteria. What is one disease caused by mycobacteria? Tuberculosis. Tuberculosis affects like 1/3 of the world's population. It's a huge problem. So we want to design antibiotics to treat tuberculosis. Just while we're on it, leprosy is also caused by mycobacteria. Do you all remember what's special about mycobacteria? what they have in their cell wall? Mycolic acid. It's a waxy substance. Are mycobacteria gram-positive, gram-negative, or neither? They are neither. They are acid-fast positive. If that was a guess, it was a good guess. All right, so we mentioned quinolones. One of them that you've seen by the name brand is ciprofloxacin. By the way, I'm going to focus on the name of the drug, not the brand name, as we go through today, because that's just, it's too much to put brand names on top of these. Okay, so quinolones, this drug that I have trouble pronouncing that starts with an I, isoniazid, I'll get it eventually, and sulfonylamides, these all inhibit cell wall synthesis. Or pardon me, cell wall synthesis, DNA synthesis. So chromosome replication. The next group is going to inhibit cell wall synthesis. What is a component of the cell wall? Let's talk gram-positive bacteria. What do gram-positive have a lot of in their cell wall? Big P word.
Is it the
Peptidoglycan.
There you go, peptidoglycan. So betalactam is this big umbrella group of cell wall synthesis inhibitors. Penicillin is a betalactam antibiotic. So betalactam is your big category. Penicillins fit within that category. And you'll notice that we have some, in this figure, we have some natural and then some semi-synthetic penicillins. Don't memorize the structure of these. This is not a chemistry class. So penicillins, these are going to be effective against both gram-positive and gram-negative bacteria. These are going to prevent peptidoglycan from forming a nice rigid cell wall. So it prevents the peptidoglycan layers from attaching to each other. Right? In gram-positives, we have a really thick wall, so that peptidoglycan doesn't form a nice cell wall. Gram-negative bacteria have a thinner peptidoglycan layer, and they have a layer of lipids. Remember the lipopolysaccharide, LPS? That's in gram-negatives. So beta-lactams, like penicillin, these are more effective against gram-positive bacteria than gram-negatives, because gram-negatives have that outer lipid layer that gram-positives don't have. So gram-negatives might not be able to synthesize their rigid part of their cell wall as easily, but they still have an outer layer that gram-positives don't have. But these are effective against both positive and negative gram species. And in case you're looking for it, your book does not go into detail about the more or less effective. That's just something that I would like for you all to know. Okay, so some people are allergic to penicillin, so that's why we're going to have these semi-synthetic penicillins. You can take the drug that somebody is allergic to, tweak it just a little bit, so now it's still effective, but the host, the human, doesn't mount an allergic response to it. We also have some penicillin-resistant species. So again, if they're resistant to a drug, we just tweak it a little bit, now it's effective, and they don't have resistance against it because it's a different molecule. So some examples are ampicillin and amoxicillin. Again, we've heard these before. We've seen these. Then just as we go down our list of other beta-lactams, cephalosporins are also effective against gram-positive and gram-negative species. As are carbapenems. I know, my head's already spinning too. So let's just try and breathe and take this slow. All of these drugs that we've talked about inhibit peptidoglycan, forming a nice rigid cell wall in bacteria, so the cells die. As they try and divide, they're going to burst open and they die. That's what all these drugs do. That's what I want you to know. You don't need to know how to spell them. All right, so instead of just flying through it, let's just revisit what we talked about. Betalactam antibiotics prevent peptidoglycan from cross-linking or forming a nice rigid layer in a cell wall. It's effective against gram-positive and gram-negative bacteria. These include penicillin, semi-synthetic penicillins, including these here: amoxicillin, methicillin, ampicillin, carbenicillin is one I've never heard of before, but we've heard of these three probably. So all of these drugs functionally do the same thing. Why do we have some that are different than others? Because of drug resistance in bacteria and human allergies. That's why we have to have a diverse array of drugs that do the same thing. Okay, so cell wall synthesis inhibition is one way to deal with bacteria. And if you take a look here, I really like this figure. I've included a set of summary figures at the end of today's presentation. So this is the group of drugs that we just talked about. And vancomycin is going to be included in there. But this is only effective against gram-positive bacteria. Vancomycin. The others were broad spectrum. Now vancomycin does the same thing, but only affects gram-positive organisms. So on this figure, maybe put a little star next to it, or next to the vancomycin, just write like G positive, or just a plus sign. And so this drug, it's within the same group of cell wall synthesis inhibitors. So you would only choose this if you absolutely had to. This is like a last resort because it has some really nasty side effects. It's hard on your kidneys. So only unless you had to would you take this drug. All right, there are two polypeptide antibiotics. A polypeptide is made up of what? Polypeptide. Peptide. What do peptides make up? Huh? Yeah, it's a chain of amino acids. It's a protein. This is a protein antibiotic. So these are going to affect the cell envelope. The cell envelope includes the plasma membrane and the cell wall. So things that separate the cytoplasm from the external environment, the cell envelope. Bacitracin is one drug. So this is going to affect the ability of bacteria to send their cell wall components through the membrane. They have to go through the membrane to build the wall. This drug prevents that from happening. So you can't build a cell wall. And so this is only used topically. You all have seen this in Neosporin. So if we put bacitracin with neomycin and polymyxin B, then it's sold under the brand name Neosporin. Polymyxins are effective against gram-negative bacteria, and they are going to increase the membrane permeability. So that's going to mean that it's going to make a cell basically leak its cytoplasm out. So you're kind of like poking holes in the membrane with a polymyxin, which is, again, keep in mind, in those topical ointments like Neosporin or polysporin. And so while we're on it, bacitracin, put a G positive next to that. Polymyxins, put a G negative next to that. So that is one reason why we're going to combine these two drugs in the topical ointment. One is effective against gram-positive, one's effective against gram-negative. You put the two together, now you've created a broad-spectrum drug from two narrow-spectrum peptide drugs. All right, let's talk about protein synthesis inhibition. What component of a cell is going to build proteins? What is our factory for protein synthesis? Yeah, the ribosome, right. So some of these drugs that are going to block ribosomes from functioning are going to be the aminoglycosides, Okay, so I think I need to adjust this slide. These slides come like pre-cooked and then I tweak them through the book, pre-cooked. These are available through the internet and then I adjust them. And I think that this is an error because ribosomes do not perform transcription as far as I'm aware, they translate. So either way, an aminoglycoside is going to attach to a ribosome, prevents proteins from being produced. Included within this are streptomycin, gentamicin, and neomycin. So I'm going to double check this after class. Aminoglycosides attach to the bacterial ribosomes, blocking translation instead of transcription. Transcription is taking the DNA and making an mRNA transcript. That transcript goes to the ribosome translates it into a protein. So this is where I just need to touch up on my biochemistry and make sure that bacteria do the same thing that humans do. Bacterial ribosomes might be different. They may perform transcription. I need to look into that personally. All right, tetracyclines. These are going to attach to transfer RNA. All right, so follow me here. We have DNA. We have our chromosome. That chromosome has a code. We're going to take that code and create transcripts. So we transcribe it. We change the language from DNA to RNA. That RNA gets sent to a ribosome. The ribosome builds proteins, right? So polypeptides, chains of amino acids. Transfer RNA is what brings in the amino acids. So if you prevent amino acids from being brought to a ribosome, you are not going to make a polypeptide. You're not going to make a protein, your cell dies. So we can target the ribosome or the thing that's going to bring the amino acid to the ribosome, the transfer RNA. Either of those prevents proteins from being formed, cell dies. Take a look at the name, tetracycline. Tetra means 4, cycle, so a circle. Take a look here. We have 1, 2, 3, 4 circular components to our molecule, or round ring-like components to our molecule. We call that ring a benzene ring. So these are dangerous because they can get rid of our intestinal gut microbiome, which is crucial for maintaining our health. So this is an oral dose. You swallow it, and then it can wipe out your gut flora. That's a problem. Can also cause your teeth to stain. I've never seen this. Anybody seen this? Stained teeth? Yeah? So I'm not sure what causes that, honestly. All right, so take a look here. So a ribosome is made up of 2 subunits. So what we've talked about so far, the aminoglycosides and tetracyclines attach to or inhibit one subunit. Chloramphenicol, the macrolides, all these other drugs are going to inhibit the other subunit. What I want you to know here is just that all of these different drugs prevent proteins from being formed. Without proteins, the cell dies. That's what I want you to know. So chloramphenicol, this is a drug that also has some nasty side effects, so we're not going to use it unless we absolutely have to. Just in case you're wondering, meningitis is an infection of the layers that protect the brain and the spinal cord. They're called meninges. If you see itis, think infection. Meningitis is an infection of the meninges. I know this is an anatomy class, but it's basically an infection of right outside the central nervous system. You got to have a brain and a spinal cord to know what's going on. You don't want that to be infected. And anemia is a lack of iron. We need iron to transport oxygen in our red blood cells. Without oxygen, we die. But I guess while we're at it, it's probably better to take a drug that keeps you alive from an infection than to worry about having low iron, right? Survival is more important than anything else. Erythromycin is only effective against gram-positives, so we would call this a narrow-spectrum drug. Clindamycin, lincosamide, streptogramins. Again, these are all drugs that I would like for you to just be moderately familiar with. Don't expect me to say, which of the following drugs goes under the brand name Zybox. Okay, I'm not going to ask you anything about brand names, I promise. And I understand that we have so much to memorize, so what I want you to see here are big picture things. Slides like this where we just have it full of drug types are less important, but I'd like for you to look over it, okay? Because again, in a healthcare setting, you're going to have a book available. You're going to have the internet available. I do want you to know how betalactams work, though. That is very important. So some drugs have been developed and are effective against bacteria that are resistant to other drugs. So MRSA, we've talked about over and over and over again. That is methicillin-resistant Staphylococcus aureus. So it's resistant to methicillin, but here we go, oxazolidinones do kill MRSA. We can inhibit RNA synthesis with drugs like rifampin. These are effective against mycobacterial species. Again, those cause tuberculosis and leprosy. But again, nasty side effects, yes.
I thought MRSA was one of those diseases that you never truly got rid of. Like you could treat the symptoms and it would lay dormant until something vamped it back up.
So as far as I know, I would have to dig in the literature to answer that question directly. According to the textbook, though, certain drugs can kill MRSA. Now, we could end up with some sort of a multi-drug resistant Staph aureus. But as far as the textbook states, this drug right here, something under the brand name like Zyvox, would be effective against MRSA.
Okay, thank you.
Methicillin would not be effective because this organism has resistance to methicillin.
Right.
But really good question, comment. I will look into that after class today.
Thank you.
Yep. All right, so I'm going to go for about 5 more minutes here. So all of those drugs that we've talked about, those are all antibacterial. Drugs that kill bacteria. That's what this class is mostly about. So I want you to focus on all of the antibacterials, all right? Antivirals, they talk a lot about HIV in this book, and I'm going to talk a little bit about HPV here in this next slide. So if we have antiviral drugs, we can either try and block the virus from getting into a cell, try and prevent the viral genome from replicating, and in that we would prevent new viruses from being produced. So you'll have heard of Gardasil. This is effective against the human papillomavirus. HPV causes almost all cases of cervical cancer in women. It's also linked to penile cancer in men as well as throat cancer. So this is a virus that causes cancer. You can take a vaccine that is effective against cancer, ultimately. And this is approved in men, too. So if you don't have it and you're of the age that can get it, look into it. This is really helpful for you and your future partners, current partners, future partners. So this works just like most vaccines. Your body recognizes a viral protein, you make antibodies, and then you're ready for it if you deal with it again in the future. Antifungal drugs, don't worry about these, honestly. Look into it if you're interested, but I would really rather you all focus your studying on antibacterial followed by antiviral drugs. Same with antiprotistins. Some things that you all have heard of are quinine and chloroquine. These are effective against protists like Plasmodium that cause malaria. So if somebody comes back from the tropics, say, and they have malaria, they might get quinine or chloroquine. Helminths are wormy parasites. Again, your book goes through all these different drugs, and for the sake of time, we're not going to go through all of these. I'd rather focus on tests. I would like for you to understand these microbial tests. Again, for the exam, don't even worry about anti-helminth and antifungal drugs. I want you to study antibacterial drugs. Okay? This is a lot to do. I'm not going to expect you to do it all. Okay, so the MIC test. You could also see this called the tube dilution test. Really straightforward. We're going to take an antibiotic, and we're going to put it in different tubes at different concentrations. So imagine we have 100% antibiotic. Cut it in half, 50%, 10%, 5%. You put bacteria in there, and you're going to see when do they stop growing. What is the lowest concentration of antibiotic that will prevent the growth? So let's say they grow at 5%, they grow at 10%, but they don't grow at 20%. You would call 20% the minimum inhibitory concentration. It's the lowest concentration that prevents the growth of bacteria. MIC. I do want you to know that. You will see this in your lab. I'm trying to make lecture correspond with lab as much as I can. You're also going to see this. We talked about this yesterday, the diffusion method. There are 2 ways to do this. One is the E-test method. So you have a strip with antibiotics from a high to a low concentration. what you're going to do is you're going to look and see where do the bacteria grow in contact with this strip. So you're going to see kind of like I would expect a V shape where high concentrations, you're going to have no growth, just like Fleming saw with the mold and the bacteria not growing. At a low concentration, you should see bacteria touching this, meaning that the antibiotic doesn't kill them. So where they stop growing is the MIC. This diffusion method, just like we saw yesterday, you're going to put different concentrations on a lawn of bacteria. Do they grow? Do they not grow? Measure how far the zone of inhibition is. And then based on the size of it, we can say that it is resistant or susceptible. Some bacteria can develop mutations spontaneously on their own. Organisms can also transfer resistance mechanisms from one bacterium to another. Don't worry about the difference between these three processes because we are not doing chapter 7 and 8. So don't worry about these three. Just know that bacteria can generate spontaneous mutations or transfer resistance to another organism. All right. I just want to see where we're at. Okay, so this is the last slide I'm going to talk about today. I'm sorry, what's up?
Can I ask you to repeat the part about the MIC one more time?
Yeah, definitely. All right, so what you want to do is you're going to expose bacteria to higher and lower concentrations. If bacteria grow, then we are going to say that they are resistant to an antibiotic. Once they get up to a higher concentration, they're going to stop growing. Whatever that concentration is, the lowest one that they don't grow is the minimum inhibitory concentration. So the concentration that prevents them from growing, but it's the lowest one. Because again, we want our therapeutic dose to be low relative to our lethal dose.
Thank you.
You're welcome. Okay, so last thing I'm going to talk about. I do want you to know this slide for your exam too. Big picture stuff, right? Offensive versus defensive mechanisms of dealing with antibiotics. Let's talk about offensive. So bacteria can break down an antibiotic. It comes in the cell, they don't like it, they destroy it. That's offensive. They can also just modify it, so they can add a phosphate group on. That's phosphorylation. And now it's a different molecule, it's not effective against them. So you can break it down or change it. Defensive strategies, they can just pump it right back out. You send it in, I'm going to get rid of it. Or they can alter their target. So let's say a drug is effective against a ribosome. Some bacteria can just mutate the ribosome shape. Now it's not effective anymore. That's a defensive strategy. So you can expect maybe me to give you one of these four scenarios and then say, is this offensive or defensive? Something like that. Or select the defensive strategy of the four options.
Can you
Something like that.
defensive again?
Yeah, so defensive, we can either just pump out the antibiotic, so it comes in the cell, you just get rid of it, or you can just alter the target. So, for example, in the figure, it shows ribosome. If an antibiotic targets a ribosome, some bacteria can mutate their ribosomes to a different shape, so now the target doesn't bind to where it should, because it just doesn't fit like that, kind of like the substrate enzyme complex that we talked about. How it's like a lock and key situation. Okay, you all. So I'm going to go ahead and stop so that we can log into our Respondus stuff. I'm going to be by my email over the next half hour. So if we have any issues, let me know. If you want to take it in here, you can. If you want to take it out in the hall, whatever you want to do, find an empty classroom, I'll be here for the next half hour. So again, for this information for the exam, you'll notice at the end of the slideshow, I have big figures. I want you to focus on the offensive, defensive, and antibiotics most. Don't worry about antifungals. Don't worry about anti-parasitic. Okay? right, so I'm going to stop this